Thank you for joining us today for part 3 of the GastroPlus lecture series, DDI Predictions with Viera Lukacova. Notes. For optimal video and audio connection, we recommend you close any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. This webinar is being recorded for future playback and on our website and YouTube channel. For the Q&A session, you may ask questions via the questions panel on your dashboard. You may submit queries at any time during the presentation. And if you need assistance, please raise your hand or use the chat function. Today's 90-minute schedule will begin with a 30-minute presentation, followed by a rapid-fire 10-minute Q&A. And in the second half, Viera will lead us through dynamic simulations, which will then finally close out with a second Q&A session. On behalf of everyone at Simulations Plus, we appreciate you spending your morning, afternoon, or evening with us. To get started and tell us a little bit about yourselves, how familiar are you with GastroPlus software? We'll give you a few seconds here to go ahead and give your feedback. It looks like most of you are somewhat familiar with our software. So today, it gives me great pleasure to introduce today's speaker, Viera Lukacova. As the Director of Simulation Sciences at Simulations Plus, Viera is described by her colleagues as resourceful, brilliant, and dedicated to advancing the science of modeling and simulation. Her clients describe her as a visionary, trustworthy, and a world leader in PBPK modeling. Scientist.com highlighted her as one of the top 19 women in STEM. And if you ever forget how to pronounce her last name, well, there's a default method for KP calculation to remind you. Please welcome my friend and PBPK diva, Viera Lukacova. Viera, take it away.
Thank you, Arlene, for the very kind introduction. And thank you all for taking time out of your busy schedules to learn a little bit about the GastroPlus and its utility in predicting drug-drug interactions. Navigate slides. So just a very brief introduction. Of course, the DDI module for predicting.
Oh, Viera, we can't see your screen.
Not at all. Okay. Can you see it now?
Yes.
Can you see the right?
Not the right one.
Good thing we practiced this.
Exactly. Well, our audience will appreciate our practice session before.
Okay, there we go. Can we see the right screen now?
Now we've got your let's not forget slide, and we'll start. Take 2. Take it away, Viera.
Perfect. Thank you, Arlene. So the DDI module within GastroPlus is, of course, linked to the entire GastroPlus absorption and PBPK model. So within the DDI predictions, all of the processes that you typically look into for in terms of drug dissolution in the intestine, absorption, and so on, are accounted for. So this does give you a good strategy for predicting the drug-drug interactions, considering that you have an accurate estimate or prediction of the drug being absorbed, drug being metabolized in the intestine, in the liver, as well as getting into the systemic circulation. And having accurate simulation or accurate model for all of these processes is really the prerequisite for having also accurate predictions for the drug-drug interactions. So let's go more in detail in the capabilities for the DDI specifically. Within the GastroPlus, you have an option to do the simpler steady state or static predictions for drug-drug interactions, all doing full dynamic simulation between the perpetrator and the victim compound. For any of these, you can account for all typical interactions, competitive inhibition, time-dependent inhibition, induction, or any combination of all of these. So one perpetrator could actually act as an inhibitor as well as inducer at the same time. And all of these combinations you can incorporate in your drug-drug interaction predictions. The DDI module, and we will discuss this a little bit more towards the end of the presentation, but the module does come with a list of, or with number of models for which we have already built PBPK, full absorption PBPK models that you could use to start predicting drug-drug interactions with your compounds. These are, on the slide, you see a snapshot of some of these models. It's not an exhaustive list. We keep updating these and adding new models with every release. And the selection is really based more on the FDA guidance of index substrates and inhibitors of enzymes and transporters. So we are trying to cover the variety of the compounds that you would be typically using in DDI studies. Now, keep in mind that this is not an exhaustive list, and this is not a limitation of the software. We are providing these more as a convenience for you so that you don't have to spend the time on building the models that we have created already. But you can use the DDI simulations in GastroPlus to predict the drug-drug interactions between any two compounds. So if you need to use an inhibitor or an inducer or a substrate that we have not provided yet, you can still do the interaction or DDI prediction for that compound. You will have to build the model yourself, but you can still do the interaction. So let's start with the simpler type of predictions with the steady state or the static predictions. And for those of you who have done any DDI predictions, you are probably familiar with the typical equation that is being used to predict the change in the exposure of the substrate or victim compound when it is administered with a perpetrator versus when it is administered alone. Typically, with this equation or with the static predictions, we are normally looking at the contribution of the intestine to the DDI and the contribution of liver or the systemic circulation. And in both of these parts, you can account for the simple competitive inhibition, time-dependent inhibition of enzyme, or induction of the enzyme. And what we are showing here is the simplified version of the equation, assuming a single perpetrator, so single inhibitor or an inducer in the system. But the way the equation is actually implemented in the program, you would be able to account for multiple perpetrators as well. For example, if the perpetrator that you are administering has metabolites which are also affecting the same enzymes, you can account for the combined effect of all of the compounds. So for it, there could be multiple competitive inhibitors, multiple time-dependent inhibitors, or multiple inducers in the system. And the equation is automatically accounting for all of those effects. So what is the type of information that you need to provide to the software for this type of steady state prediction? For the victim compound, it is the FM values, which is contribution of each enzyme to the clearance or elimination of the victim compound. Which enzyme is contributing, how much it is contributing. In addition, to correctly account for the effect of intestine on the drug-drug interaction, the additional parameter and this fraction of the compound which escapes intestinal metabolism is also important. And we'll show you how you could use built-in tool within GastroPlus to estimate the FM values from in vitro experiments that you might be performing more in the early stages of the drug development. For the inhibitor or perpetrator, there is more extensive information that is required. One is defining the parameters, defining the interaction, whether it is Ki or IC50 or EC50 and Emax for the inducer, and so on. And finally, the equation requires effective concentration of the inhibitor or inducer in the intestine and in the liver or systemic circulation. And this is where program or GastroPlus allows you to select from a number of different options for estimating these concentrations for the inhibitor and inducer. And we will discuss a little bit what choices you need to make depending on what kind of interaction parameters or constants you have. And we'll be going through the list of these, how you could define all of the parameters within the program through an example. So let's say you have a compound. Your compound in the development is metabolized or eliminated mainly by CYP enzymes. And to make it a little bit more complex, we've created a list of several different CYP enzymes that might be contributing to the clearance of this compound. And you want to evaluate what would be a potential drug-drug interaction of this compound when it is administered with inhibitors of the CYP enzymes. Is that something that you would have to worry about once the compound gets into the clinic? So for this, we will use the GastroPlus, and I have GastroPlus running here. So the first step I would do is create a new database where I will start entering the properties or the information that was measured in vitro. I'll create new database. And since I'm going to be working with simple static prediction, in this case, I'm not worried about the absorption parameters for the victim compound. I'm going to go directly into the DDI module. And to give you a very quick overview of what the DDI module looks like, you can see it is split into two sections. The left side of the window corresponds to the current compound, which means the compound for which you want to do the predictions or the record from the GastroPlus database from which you opened the DDI module. The right side of the window is dedicated to interacting compound. And this could be coming from the database of standards that we are providing. or could be coming from your own database. If you have your own model for the interacting compound, you could simply go under File, Open Database, and open any other GastroPlus database with your models. For the current compound, you designate this compound to be either perpetrator or a victim of the interaction. And the interacting compound is automatically going to be designated as its counterpart. So if my current compound is a victim, the interacting compound will be a perpetrator. I'm going to keep it with this setting for this example, since we are going to evaluate our current compound as a potential victim in the interaction with inhibitors. And this is just the smallest screenshot of that slide behind, so that I have easy visibility for all the in vitro experiments that were done, that I will use to calculate the contributions of individual enzymes to the clearance of our victim compound. Okay. So again, for the static predictions, the main information that you need to provide is the FM values for the victim compound, meaning how much each enzyme is contributing to the metabolism of that compound. And if you already have that information, if you've done the calculation, you can simply use these buttons at the bottom to define it manually. So if I click Add Enzyme or Transporter, I could simply select what enzyme it is and define a contribution if I already know. And I could add another enzyme. Oops. And define these contributions. Oh, come on. For some reason, okay. Define the contributions of FM values. Typically, you would obtain these FM values from in vitro experiments either by using recombinant systems or by using human liver microsomes and measuring the clearance of your compound in the presence of inhibitors, of specific inhibitors of individual enzymes. And if you have that kind of in vitro information measured, you could use a calculator built into the DDI module to estimate the FM values directly from your raw data that you measured in vitro. So I will demonstrate quickly how you could easily use that data within the calculator to estimate your FM values. I'm going to click on Calculate FM Values. And again, we have two sections here, depending on what kind of in vitro assay you used. Whether you used recombinant enzymes or microsomes, you would select the appropriate tab. In this case, our in vitro data is coming from microsomal assay, where several sets of experiments were performed. One was measuring intrinsic clearance without any inhibition. So the total intrinsic clearance for this compound was measured as 269 microliters per minute per milligram protein. So I will enter that total clearance in here in this window as the total clearance for the compound. And now the additional set of experiments was performed by measuring the clearance of our compound in the presence of 4 different inhibitors of specific enzymes. So we use the naftoflavone for 1A2, fluconazole to estimate the 2C19, quinidine as an inhibitor of 2D6, and finally ketoconazole as an inhibitor of 3A4. And these are the individual clearances that were measured in those experiments. So I would enter those individual clearances measured in the presence of inhibitor of specific enzyme in this table up here. So inhibiting 1A2, our clearance, the measured clearance was 240. Inhibiting 2C9, the measured clearance was 250. Inhibiting 2D6, the clearance went down to 200. And finally, inhibiting 3A4, the clearance went down to 100. The additional piece of information that you need to provide is what was the amount of enzyme, CYP enzyme, in the specific microsomal preparation that you've used. The program does have built-in average expression levels in the population of subjects, and you could select Caucasian, Japanese, or Chinese population. But depending on what was the source of your microsomes for the in vitro data, the actual amount of the enzymes in your in vitro preparation might be different. And if it is important to account for the differences between that preparation and what would be the average expression in vivo. So in this case, we had the microsomal preparation with 42 picomoles of 1A2, 32 picomoles of 2C9, 9.1 picomoles of 2D6, and finally 78 picomoles of 3A4. So based on all of this information, the program now has enough information to estimate what would be the contributions of individual enzymes. And for easier visualization, the plot also represents the individual enzymes. So majority would be contributed by 3A4, with smaller contributions from 2D6, 2C9, and 1A2. The additional aspect that you can account for is if you have maybe from animal experiments or from renal secretion information or expectation of additional type of clearance, you can account for other type of clearance in the enzyme, in vivo. So for this case, we are assuming that maybe 10% of the clearance would be coming from other sources, which means that clearance is not going to be inhibited or by CYP inhibitors in vivo. And again, the program recalculated the contributions of the other CYP enzymes by accounting for that contribution of other clearance. Once I have the calculation done, if I click export FM values, the program will send these final contributions back into our main table in the DDI module. So I can close this calculator, and the program translated here or saved here the FM values. To help you remember how these FM values were obtained, the program puts here source. I cannot modify. The program is telling me that I've used the HLM calculator, so my data is coming from the HLM. And I can now come and save the information for subsequent uses at the later time as well. In this example, I'm going to look specifically at the contribution of the liver to the DDI. But with 3A4, it is possible that the drug-drug interactions or the interaction in the intestine will also be a significant contribution. So if I wanted, I could add additional enzyme 3A4 for the gut at high contribution, since that is one of the major enzymes in the intestine, but would also need to define the amount that escapes the gut metabolism to properly account for that interaction. To give you a little bit more background on how that conversion was done for the FM values, if you've used the recombinant systems and we're measuring clearances in the individual RCIP systems, the additional calculation the program would do would be intersystem extrapolation factors, and from those calculating the clearance for each CYP enzyme, and finally the FM values. And this is coming from previously published, so standard equation here. The ones that we've used were based on the HLM measurements. So again, from the differences between the total HLM clearance and the clearance measured in the presence of a specific inhibitor, we can estimate what would be the FM values for the individual CYP enzymes. I'm not going to spend too much time on the equations. You will have access to these slides and you are welcome to study them in detail at a later time. So the next step to define is the interaction or the inhibitor or inducer type parameters. Coming back to the DDI module, our interaction parameters would be defined for the perpetrator. And we'll be doing, for this example, let's try to do one of the simpler ones with a fluconazole. And based on the information from literature, fluconazole can actually inhibit several different CYP enzymes, 2C9, 2C19, and 3A4, as well as 3A5. So it could inhibit several of the enzymes that are metabolizing our compound, which means we need to make sure that we are accounting for or defining the interaction parameters for all of those enzymes. Now, the first step that you need to define is what type of interaction you are dealing with. And this is where we'll spend a little bit more time on this specific column in the perpetrator parameters table, which is called inhibitor or inducer constant type. And you see these are fairly cryptic names, but there is a reason behind that madness. That name does tell the program what type of interaction it is dealing with, what was the source of the data, as well as whether the value is corrected for non-specific binding. So are we using unbound value or a total value? I'll switch here to the next slide where it is broken down into individual parts. So the very first part of that name, KI, IC50, or EC50, tells the program whether this is an inhibition. KI or IC50 means it is an inhibitor. EC50 means it is an inducer. The second part of that name, the REV or could be IRR, is applicable only for inhibitors. REV stands for reversible or competitive inhibition. IRR is used for irreversible or time-dependent inhibition. So the combination of the first two parts tells the program whether it is competitive inhibitor, time-dependent inhibitor, or an inducer for the interaction. The third part, where you are defining in vitro or in vivo, talks about the source of the measurement. The in vitro measurements would typically be, at least for enzymes, would typically be coming from microsomes or recombinant enzymes. So would be more related to the concentration of the compound directly at the site of metabolism. The in vivo means it was estimated based on the clinical study data and typically would be related to the concentration of the compound in plasma. And depending on the type of compound and what distribution mechanisms are relevant for that compound, the concentration, at least the unbound concentration in plasma and in a specific tissue might be the same. So in those cases, the in vitro or in vivo unbound value would be the same. But it is not the rule. For some compounds, especially if the transporters are involved, there could be a significant difference between the concentration in plasma and the concentration in a specific tissue. So that would affect how you use this Ki or EC50 value. And finally, the last part of the name, which you probably guessed already, U stands for unbound or corrected for the non-specific binding in the measurement environment, and T means total, so not corrected for the non-specific binding. From our experience, the ideal measurements are the unbound in vitro measurements, so representing the concentration at the site of metabolism, and unbound Ki or EC50 values. So for our example, we have already entered these values in the database. If you are working on your own compound, you can easily add these values. If you simply click Add Enzyme or Transporter, define what enzyme or transporter it represents, what was the type of parameter, and define the values for the parameter. Keep in mind for induction, so if it is EC50 value, in addition to EC50 value, you need to define also Emax, which would be defined in this column. So let's say it would be Emax of 10. Similar for time-dependent inhibitor. So if I select it here, Ki, IRR, irreversible or time-dependent. In that case, I need to define the K inact value, right? And that one would be entered in the same column. So K inact, let's say, would be 0.012. So both K inact and Emax values are entered in the same column, and depending on the interaction constant type, the program knows the meaning of that parameter. For the simple competitive Ki values, that column is disabled because you don't need to define additional parameter for that. So for now, we are trying to do interaction with our compound, which is metabolized by 1A2, 2C9, 2D6, and 3A4. The overlapping enzyme with the perpetrator are 2C9 and 3A4, so we need to select parameters for those. For 2C9, we do have information for unbound in vitro value, so I would select that one. For 3A4, again, from literature, we found several different parameter values. I would recommend using the in vitro parameter values. We don't have any unbound, so I would select one of the total ones. And you can see there was a number of different values that were reported in literature. If you want to look at more conservative estimate, of course, you would be selecting the lowest possible value. And that would be my estimate if I'm trying to do a prediction a priori. So that would be for defining the interaction constants for the perpetrator. And finally, the last type of information that we need for the perpetrator is what concentration we want to use. And if you've read any publications on doing the steady state or static predictions, you might have seen different recommendations on what types of values should be used. Should we be using maximum plasma concentration, liver inlet concentration, or concentration in the liver tissue, should these be total or unbound concentrations, and so on. If you read 5 different papers, you'll likely get 5 different recommendations. So we have provided all of these options. And place where you would be selecting the concentrations is this button down here, perpetrator steady state concentrations. This is where you can select from a number of different concentrations, calculated or simulated. So what do these mean? I'm going to clean up my screen a little bit. The first section, the calculated steady state concentrations, refers to the standard pharmacokinetic calculations, assuming steady state has been reached. And it is simply based on the dosing regimen, clearance of the compound, bioavailability, and so on. So these equations refer to the concentrations in this section. And I'm going to randomly select a few of those. The second section in that window refers to simulated concentrations. This is something that you could use in cases where a full PK model is available for the perpetrator compound. For example, if you are using one of the perpetrators from our database of standards, the PK models are available. So you could run simulation at least for the perpetrator and estimate the concentration from the simulated concentrations. And depending on when the substrate or victim was administered, the perpetrator could already be in steady state, in which case the effective concentration would be the average concentration. If the steady state has not been reached yet, then the effective concentration for the perpetrator is estimated from the AUC across the dosing interval at the time of the administration of the victim compound. And if you are accounting also for intestinal interaction, the simulated effective concentration for the gut is dependent on the transit of the compound through the intestine and is estimated as the average of the enterocyte concentrations at the time when the victim compound reaches each individual intestinal region. Okay. So in here, let me select some of the simulated concentrations as well. And when I click OK, because I selected some simulated concentrations, the program is running the simulation in the background. And we have predictions for the DDI shown down here. And it's kind of a mess because there are a few settings that I haven't done. In this case on purpose, so that I can demonstrate what could happen, right? With the use, you will be avoiding these mistakes as you are using the software more and more. So the first thing that you might have noticed is this warning label here in the corner, warning you about the mismatch in the Ki and concentration type. And it also tells you, if you click here, the program does give you explanation on what the mismatch actually means. As you recall, I've spent quite a bit of time describing the name for the interaction constant. And it is important in terms of how you are linking the interaction constant with the concentration of the perpetrator. So one of these aspects that the program is looking at should be pretty clear. If you are using unbound Ki value, you should be using unbound concentration for the perpetrator. If you are using total, then you should be using total concentration for the perpetrator. I think that one is pretty obvious. The other one is if you are using the in vitro measured Ki values, this represents the concentration at the site of metabolism, which means you should be pairing that or linking that with a liver concentration or tissue concentration. If you are using Ki value that was based on plasma concentrations from clinical study, then of course you should be linking that with a plasma concentration for your perpetrator. So that is what the program is warning me about, that I don't have these connections or these matches correctly matched. And to make it easier, the program also marks the concentration types which are not matched well with our inhibitor concentrations or types in red. Right? And right now they are all red. So the first thing that would be easy to fix, right, is dealing with the unbound and total Ki values. If you don't have unbound value, you have a total value, you can still tell the program how to convert it into unbound value. So in here, under in vitro FU type, I can select to either define the FU directly or have it estimated from the compound properties. In this case, I'm going to use one of the established methods for estimating the fraction unbound in microsomes. It's published method by Halifax, which would estimate that in a typical assay with the 5 milligram per milliliter microsomal protein, the fraction unbound would actually be fairly high. And now the program can use it to convert or to recalculate the unbound Ki value from the total. And you will see that change resulted in 2 of my entries being now marked in black, because now we have a correct match and I can use the liver unbound concentrations, whether it's calculated or the simulated one for prediction. The others refer either to total concentration or the plasma concentration. So those are still out of sync with the inhibition constants, and those would not be recommended to be used. So you can either ignore those results in this setting, or if you want to have a cleaner results, you can come here and make sure to use only the correct concentrations to keep only the marked values. Now you will see we have a prediction for the simulated liver concentration. We don't have prediction for the liver inlet. That is based on these standard equations. And one parameter that is important is a dosing interval. And again, we've tried to put as much warnings in this module as possible. You can see the dosing interval is actually highlighted in yellow, the program drawing your attention to that entry that you did not provide the value, what would be the dosing interval. So let's say we'll do 24. And now we have prediction for both the liver inlet concentration calculated from the typical equations, right, assuming steady state has been reached, and the simulated liver concentration from the model. And you can see there is a difference in the prediction. In this case, the difference is actually easily explained. It is because the calculated concentration assumes the steady state has been reached already, but the simulated concentration is based on when the substrate was actually administered. And in this specific case, we simply told the program the compounds will be co-administered on day 1. So the steady state in the simulation likely was not reached yet. Let's do a more typical design and assume that the substrate is, let's say, administered 1 hour after perpetrator on day 4 dosing. I'll simply click here to update those simulated concentrations for my perpetrator. And you will see now the predictions are much closer to each other because the steady state or the perpetrator is getting closer to steady state even in the simulation. You can see what the difference would be if you click on show plot. And this is showing the unbound liver concentration, which I'm using, the simulated one. In this case, the concentration is estimated from the simulated concentration in this range. The very first prediction was based on the first dose. So there the inhibitor concentration would be much lower and resulted in lower DDI prediction. And I think this covered just about everything we can do with these steady state predictions. And we can take a quick break from the presentation and address some of the questions that you might have posted already in the questions panel.
Thanks, Viera. We are off to a great start and several questions are in the panel. While Yuzhuan gets ready to start the first one, we'll quickly ask the audience, what platform are you using to develop your PBBM and PBPK models? Feel free to choose all options that apply. And keep those questions coming in. You can go ahead and submit them again through the questions panel on your dashboard. Just a few more seconds here. Well, I'd also like to introduce the moderator for our first Q&A session. Yuzhuan Zhang is a highly skilled scientist on the simulation studies team, and tonight she's taking a break from building PBPK models for small molecules with the dermal administration route to step in and coordinate your questions for Viera. The floor is yours, Yuzhuan.
Hi, everyone. Thank you for taking your time to attend this seminar. And here is a question for the first section. Some of the question audience mentioned they already have the answer, just in case other audience has a similar question, and also Viera has more comments. So first question is, in the calculation of FM from the human liver microsome for the intrinsic clearance values, do you take into account the concentration at which this was measured? 3A4 will always be the most important at high concentration since it is a high capacity enzyme, but at lower, maybe at possibly the clinical relevant or other enzyme, maybe the major. How do you do the calculation of the FM?
So that simple calculator does not really look much at the concentrations at which, or does not scale the clearances for the concentrations at which they were measured. Right? So in this case, it would be important to try to do the measurements at the clinically relevant concentrations. Right? So if the clinically relevant concentration is lower concentration, then it should be done for all of those enzymes at the lower concentration so that these dependencies do come, are correctly accounted for. Of course, the other option would be going to a full dynamic simulation, which we will go through in a short while, in which case the Km values, Vmax values for the individual enzymes would be able to better distinguish how DDI risk might be changing, let's say if you are changing a dose within the clinical study.
Okay, thank you, Viera. And another question also about how to calculate the FM by microsome. Should I use each specific inhibitor for enzyme?
Yes, if you are using the microsomes for these measurements, then you need to do measurement of one clearance without an inhibition, so get the total clearance. And then you would have to do measurements in the presence of specific inhibitors of individual enzymes, one at a time, right? So that you always inhibit only one enzyme, and then from the difference of the uninhibited clearance and clearance inhibited by that specific enzyme would give you better information on that enzyme itself. Of course, the other option is to try to do measurements from the recombinance of enzymes, where you don't have to rely on a specific inhibitor because you would be measuring only contribution of specific pathway.
Okay, yeah, thank you, Viera. It's very insightful. I think there's no more other questions.
Okay, so if there are no other questions on this part, we'll proceed into the dynamic simulations. The dynamic simulations do give you more flexibility and avoid some of the assumptions and simplifications in the steady state type predictions, because these now allow to account for interaction in any tissue, especially if you are using PBPK model to describe the PK of your compound. You can account for the interactions in any tissue that might be expressing the enzyme of interest. It accounts also for competition between multiple substrates and the inhibitors. Accounts for possibility of perpetrator acting as inhibitor inducer at the same time. This is something that you could do with steady state as well, so that carries over into dynamic simulations. You can also account for autoinduction and autoinhibition. So in this case, you are not making simplifications or assumptions on the inhibitor or inducer getting into steady state, but you can account for the effects of inhibitor's clearance changing due to autoinduction or autoinhibition effects. The equations that we are using here are, again, very standard equations. If it is competitive inhibition, we are looking at the effect on the Km value of the substrate. And it is more complex equation here because, again, we try to highlight the potential contribution or the effect of multiple substrates or inhibitors of the same enzyme. With time-dependent inhibition or induction, you would be looking more at the changes in enzyme activity. And the changes in enzyme activity would be either time-dependent inhibition due to inhibitor or induction of the enzyme. And of course, there is also recovery of the enzyme, how it is being resynthesized and recovered back in the as the inhibitor or inducer is disappearing from the system. And again, even with this, we can account for potential effect of multiple time-dependent inhibitors or multiple inducers on the activity of the enzyme. So what are the required inputs that you need for the dynamic DDI simulations? This is the list that we were looking at before with the steady state simulations. So we'll look at how this list is going to change when we are using dynamic DDI simulations. The prerequisite for doing dynamic DDI simulation is having a full PK model for perpetrator as well as victim compound. This could be either compartmental PK or PBPK, so you could use both models. The important aspect is you need to use the same type for both. So you would either be using compartmental PK for perpetrator and compartmental PK for victim, or you would be using full PBPK for perpetrator as well as full PBPK for victim. And since we have a full PK model for each of the compounds, we no longer need the FM or FG value for the victim. The full PK model for the victim compound already knows how it is being metabolized, how much each enzyme is contributing, how much a transporter might be contributing, what is the effect of the intestine. So these parameters are not important anymore. They are already included within your model. Similar to the inhibitor and inducer concentrations. That thinking about which concentration for the inhibitor or inducer you should be using, should it be plasma or liver concentration, that decision goes away because the model will automatically use the unbound concentration at the site of action. It will automatically use unbound concentration in the tissue where the metabolism is happening. For uptake transporters, it would be using unbound concentration in extracellular space. For efflux transporters, it would be using unbound concentration intracellular, because that would be the effective concentration for inhibiting the transporter. So the program automatically selects the unbound concentration at the site of action. And that means the best input for your Ki, IC50, EC50 values would really be the unbound values, right? Because they are being paired with unbound concentrations, and the values coming from in vitro assays or from assays that represent the concentration at the site of action. Okay. So we'll jump straight into an example for this. If I go back to GastroPlus, I'll close this window, and I will borrow our database of standards. As I mentioned, we do provide this DDI standards database with some of the built-in models that we've already created. And you can see it is already quite impressive list. And you would see that for number of compounds, you would see their models marked comp PK or PBPK, so that you have an option to use it with your compound, whether it has compartmental PK model or PBPK model. And in this case, we are going to try to do prediction of midazolam, which is 3A4 substrate, with ketoconazole 3A4 inhibitor. So I will select my compound. Since we do have built-in models for both of these, I have the luxury of simply using those two compounds. And I'll find midazolam, PBPK model. And I forgot to list it in here, but to demonstrate one more aspect, let me change the physiology slightly. So let's say our study, instead of using the standard 30-year-old 70-kilogram American male, maybe we want to do a simulation in females. We'll do 23 years old female physiology. So this will determine what DDI prediction or what physiology is going to be used for DDI predictions. Once I have my victim compound, and again, this could be any database. I'm just borrowing the DDI standards, but this could be any of your GastroPlus databases. And I'll open the DDI module. And we want to do interaction with a itraconazole. So we'll select with ketoconazole. We also do have a itraconazole model, but those simulations run a little bit longer. That's why I want to use ketoconazole in this case, simpler molecule. Okay. And in this case, we are going to do a full dynamic simulation. Okay. So on the top here, under the prediction type, instead of steady state prediction, I will select the dynamic simulation. And you will see when I select the dynamic simulation, number of controls will be disabled. The table with FM values is disabled. The program already knows what the FM values are based on the Km Vmax values clearance defined in our midazolam model. For ketoconazole, again, the concentration selections are disabled because the program will be automatically using the unbound ketoconazole concentration in every tissue where the metabolism is happening. And in this case, for 3A4, it would be liver and gut. So all I need to do is select which value I want to use, what interaction constant I want to use. I would select the in vitro unbound. So I'll select that one in here. What is the dosing regimen? And we'll do a typical type study where perpetrator is given once a day, and inhibitor and victim compound is administered maybe 1 hour or 2 hours after the perpetrator on day 4. Right? This is fairly standard type of study. Okay? And in this case, you can also define the dose for the substrate. It will become a more important parameter following back to the previous question, where if you have multiple enzymes and their significance or importance is changing with the concentration, then the dose would become a more important parameter to be able to define how much each enzyme is contributing. So all I need to do here is run 2 simulations at the baseline, which means the program will run simulation without interaction. So basically giving me result of what would be the result of the placebo arm. And you can see what I immediately got was a message that we have 2 different physiologies assigned to each of these records. For midazolam, I assigned a physiology for a 23-year-old female. But all the models in the database standards have the 30-year-old American male. So if you want to have interaction, they both have to be in the same body. And in case of these differences, the current compound is the one that drives the physiology that should be used. So that is all this message is telling you, it will use the physiology that was assigned to current compound. So our DDI will be based on a physiology for a 23-year-old female. It will be taking the properties of the interacting compound, but will be using them within the physiology of the current compound. This will take a few seconds to finish up. You can see the simulation does appear to be a little bit slower than what we had earlier with the fluconazole, but that is because now the program is running 2 PBPK simulations at the same time, right? Ketoconazole and midazolam. And actually this will be finished soon. Okay, so this is a prediction for the baseline simulation or for placebo arm. In the results table, the program shows the standard simulation outputs in GastroPlus, fraction absorbed, dose to portal vein, bioavailability, Cmax, Tmax, and AUC for all the compounds in the simulation. And on the plot, you can see the simulated concentrations. I'll put it on the log scale because due to the dose and exposure, the ketoconazole concentration shown in purple is much higher than the midazolam concentration in red. So ketoconazole was administered once a day, and on day 4, 1 day after ketoconazole, midazolam was administered as well. If we want to do the DDI, now the second simulation will be running full simulation. Again, agree that we are using the physiology from the current compound. And while this is running, I'll go through a couple of slides here. One was showing that these types of interactions or using the simulations that account for DDI may actually be important also for accounting for interactions between parent compound and metabolites. This is one of the cases for omeprazole metabolism, and it does have fairly complex metabolism, a lot of it being carried by CYP enzymes 2C19, 3A4. The two major metabolites, the two major primary metabolites are hydroxy and omeprazole sulfone. The hydroxy being formed mainly by 2C19, but omeprazole sulfone is subsequently also metabolized by 2C19. So once this compound, this metabolite starts accumulating, it will start competing with the parent compound. So they will be inhibiting each other. The other way around, the sulfone is metabolized mainly by 3A4, but then omeprazole sulfone is being formed mainly by 3A4, but the hydroxyomeprazole is also metabolized by 3A4. So again, once this accumulates, it will start competing with parent compound for 3A4, affecting the exposure. And this was important when we were trying to build a model, trying to match the PK or the exposure for the parent compound, as well as for both metabolites. When we did not account for the DDI, we were always dealing with slight underprediction on the parent compound as well as on the metabolite. But once we accounted for the interaction, now we are nicely matching both the parent as well as the metabolite compound. This slide is showing just a little bit of validation on one of the standard models, itraconazole. Itraconazole is a important inhibitor, one of the standard 3A4 inhibitors, index inhibitors being used in the studies. And the complication with this compound is that parent compound, as well as its 3 metabolites, are all metabolized by 3A4 and are all also inhibitors of 3A4. So to account for the DDI with other compounds, you really need to account for the effects of all 4 compounds on the substrates. And this is showing a validation across different types of studies and administration dosing regimens of the itraconazole model with the midazolam. The blue is showing observed DDI from a variety of different studies, and the red are showing the predictions from our model based on the PBPK models for itraconazole and midazolam. I believe in the meantime, the simulation has finished. So when the simulation finished, the program will add to the table the results of the full dynamic simulation. So we now have the results with the DDI, how the all of the parameters have changed. And once the program has the information for the baseline, so the placebo arm, as well as the full interaction, it automatically calculates the ratios of all of these parameters. So you can quickly see that the total effect for this dose of midazolam and this administration of ketoconazole, it would result in about 8-fold higher exposure of midazolam. And actually, a good portion of it would be coming from the intestine. About 2.5-fold is due to intestinal contribution alone, and the rest is coming from the systemic circulation. So if you look at the plot window and put it on the log scale again, do I still have showing the plot windows side by side, and I may need to modify one of these scales. So that the left axis are on similar scale, and you can now see the difference in PK in midazolam when it's administered alone versus when it's administered with ketoconazole. So to wrap up this presentation, I have only a few slides on the showing the information on the use of the models. And this is actually a publication from a group using GastroPlus. They came up with a very nice strategy for early evaluations, early risk assessment for time-dependent inhibition. Came up with a nice strategy and decision tree on how to use the simulations and early predictions based on in vitro assays to help you decide or evaluate the risk for the compound to actually be a time-dependent inhibitor. And it could help you also in the early stages in deciding selecting the compound. The applications that you all are more familiar with would be the applications in the regulatory interactions. Right? And these are actually screenshots or snapshots from a presentation that was recently a few months ago, delivered by FDA for the updated or final guidance released in 2020, in January 2020. And in this presentation, they did present the PBPK models and DDI studies. The in silico DDI studies can replace some clinical studies. The important aspect is make sure to verify the models and to have properly validated models for both the victim and the perpetrator compound. An important aspect to remember is that this is an evolving science. This is application where new uses are being considered on a daily basis. So something that was not acceptable yesterday may become acceptable next month once the reviewers or the regulatory agency gets more confidence in the accuracy of the new applications and predictions. And also in that guidance from January 2020, which was the final guidance, they provided this decision tree on how to go about doing the dynamic simulations to explore or how to use the PBPK models to evaluating the DDI potential. And you can see they also suggested that for the operating inhibitor or inducer concentrations should be using the unbound target tissue concentrations, something that GastroPlus dynamic simulation is doing automatically already. And finally, for the just a little bit, couple of comments on the validation of the models. Make sure that the PBPK model, whether it is a victim compound or a perpetrator, is correctly describing the PK of the compound for different dosing regimens, different dosing routes, whatever clinical data you have for your compound, make sure that the PK model is correctly describing that data. If it is a substrate, make sure it correctly quantitatively captures the elimination pathways, contributions of the enzymes, potential urinary secretion, and so on. And of course, the interacting compound, whether it is an index perpetrator or index substrate, also needs to be verified and need to show to the regulatory agency that these models are in fact predictive and are able to properly evaluate the effect of drug-drug interactions. And the final questions, yes, GastroPlus is also accepted as for the DDI applications. This is a snapshot of some of the recent drug product applications or approvals that were supported by GastroPlus simulations. It does have a variety of applications, but you can see that the drug-drug interactions, metabolism, transporter-based drug-drug interactions are among these accepted applications. So that brings us to our final Q&A, or maybe first a final poll from Arlene.
Thanks, Viera. That was a great list that you showed. And as a reminder, the presentation slides will be available for download from our website. The final poll question, based on what you've heard today, your interest in using GastroPlus for DDI predictions are, and you can choose any of the options that apply to you. We'll give you a few seconds to answer while Yuzhuan gets the final questions for today's seminar and lecture series prepared. All right, Yuzhuan, I'm going to go ahead and turn it over to you.
Okay, thank you, Arlene. Thank you, Viera, for the very helpful seminar. And here's a question for the second part. It's mainly about transporter. Does GastroPlus support the prediction of interactions due to the induction inhibition of drug transporters? And do reliable transporter rates of important drug transporters available?
Yes, those are very, very good questions. So in this presentation, I was focusing only on the metabolic drug-drug interactions, but yes, the transporter-based interactions can also be accounted for. I can show very briefly just one of the posters that was actually presented. Oops, this is the right one. It was, the work was done and presented by one of our colleagues back in 2012, looking at erythromycin, which is actually a inhibitor of 3A4 as well as PGP. And she has built and shown the validation of the model for both interactions, looking at the talinolol and digoxin as PGP substrates, midazolam as 3A4 substrates. Right. So yes, the transporter-based interactions can be accounted for as well, using the same equations as for the metabolic interactions. Right? The metabolism as well as the transporter aspects are all handled using Michaelis-Menten equations within GastroPlus. So the same equation would apply, of course, using the proper concentrations for the perpetrator compound. The last question on the turnover rates. Here, to make sure that everybody is on the same page as to what we are talking about with the turnover rates. It is related to the KDEC value, related to the recovery of the enzyme or transporter. Currently, we do not have specific KDEG values for the transporters. At the moment, we are assuming similar KDEG value as for CYP3A4, the half-life about 30 hours. But that is correct. It is one of the aspects that still needs to be researched more. We just haven't seen reliable values in the literature at this point.
Thank you, Viera. So another question is about the first section. What is the in vivo Ki, and how do we usually obtain the in vitro Ki values?
So you will not have in vivo Ki very often. It is something that we've added because we have seen it in some of the publications when we were looking for the Ki values for some of our perpetrators. And it basically represents value based on clinical study, based on concentrations measured in plasma and the degree of inhibition in vivo. So you will not have it very often. I doubt you would be often doing a clinical study to determine the Ki value, but we have seen it in literature for a couple of studies. So that's why we have incorporated those in the program.
Okay, thank you, Viera. I think this topic is very popular and a lot of people ask questions. So I will select the most common question first. If we have more time, we will continue discuss. And here's the next question. Can GastroPlus account for drug saturation of plasma protein binding?
That is a very good question. Not at the moment. At the moment, the PBPK model is assuming a quick equilibrium for binding to plasma proteins. So it's based on FUP alone, and it is not saturable, or kinetics of the binding is not accounted for at the moment. It is something that would be coming in one of the future versions. But it is not incorporated at the moment. So right now, if you are dealing with a compound where the saturable protein binding is an issue, you would have to select one FUP value related to the concentration that's most relevant, that is most clinically relevant. But it is something that will be coming in the future.
Thank you, Viera. So the next question is, how does the program simulate each tissue concentration of victim and inhibitor? Are they based on the plasma blood concentration?
It is standard output of the PBPK models. So within the simulation, the program, if you are using PBPK model, the concentrations for each of the tissue are calculated and simulated, and you have an option to view those concentrations. Let's say we do the ketoconazole, the one that you would be more interested in would be the liver unbound concentration, right, as the effective one. Or you do have option to look at the different concentrations, unbound and total concentrations. And depending on what mechanisms are driving the drug distribution into the tissues, it would be dependent. The concentrations in tissues would be dependent either on the Kp values as the simplest mechanism for the partitioning between the plasma and the tissue, right? Or you could have more complex mechanisms accounting for a slow diffusion through the membranes, or potentially transporter effects, transporter interactions affecting the drug getting in and out of the cells in the tissue. So all of those mechanisms you can incorporate in the PBPK model, and it is only dependent on the type of compound that you are working with.
Thank you, Viera. Next question. Are applications of DDI prediction for transporters based on interaction? Is the accuracy comparable to the metabolic interactions?
So for the properly calibrated model, right? That's the goal, to have the same accuracy before we trust the model, right? So that is the goal, to make sure that the interaction or that the prediction has the same quality. Of course, it is more difficult to parameterize and validate the baseline model, right, to account for those transporter aspects. So it does require more in vivo data, especially because for transporters, it is still more difficult to get the direct in vitro to in vivo extrapolation of the transporter effect on the compound, right? So calibrating those models with transporters usually requires more in vivo data and fitting either Km or Vmax values against in vivo data. But that is the goal, to have the same quality of the prediction, and that is where we do consider the model to be valid for either perpetrator or a victim compound, when the quality of the prediction is accurate as for the enzymes.
Okay, thank you, Viera. Another question. Can we predict the MAD exposure from SAD data if the drug inhibits itself? So it's an auto-inhibition.
It is more, so you can account for the auto-inhibition, right? But it does get difficult to predict it from single dose going into multiple doses, because single dose typically does not have significant contribution or significant aspect of the auto-inhibition. So the auto-inhibition or induction would typically take several doses before it starts manifesting itself. So, and the single dose simply would not have that information. So you would want to have at least one multiple dose study to be able to make sure that, let's say, the interaction parameters that you measured in vitro are really predictive of in vivo, to properly calibrate the time-dependent inhibition or induction effect.
Okay, there's no more other questions. I think we have 2 more minutes.
Okay, well, thank you, Viera and Yujuan, and thank you to our audience for joining us today. To evaluate GastroPlus for yourself or your team, please visit our website at www.simulations-plus.com. I predict we'll have more good questions on today's topic on our LinkedIn page. If we ran out of time to answer your questions, as I see a few more are still coming in, you can check out the ones posted on our LinkedIn page. Again, thank you for joining us, and we look forward to seeing you at our next virtual event. Have a great day. Thank you.